G protein functions as a molecular switch that is either on or off. The receptor and G protein work together with another protein, usually an enzyme. When the appropriate signaling molecule binds to the extracellular side of the receptor is activated and changes shape. Its cytoplasmic side then binds an inactive G protein, causing a GTP to displace the GDP. This activates the G protein. The activated G protein then binds to and activates phospholipase C. Once it activates the enzyme, the G protein then hydrolyzes its bound GTP to GDP and a phosphate. Phospholipase C cleaves a plasma membrane phospholipid called PIP2 into DAG and IP3. IP3 quickly diffuses through the cytosol and binds to an IP3-gated calcium channel in the ER membrane, causing it to open. Calcium ions flow out of the ER down their concentration gradient, raising the calcium ion level in the cytosol. Now remember that signal pathways are very complex. This is only one type of signal transduction pathway that may be activated by a G protein. G proteins also activate pathways that involve adenylyl cyclase CAMP and protein kinase cascades. Before the signaling molecule binds, the receptors exist as individual units referred to as monomers. The binding of a signaling molecule causes two receptor monomers to form a complex known as a dimer, in a process called dimerization. Dimerization activates the tyrosine kinase region of each monomer. Each tyrosine kinase adds a phosphate from an ATP molecule to a tyrosine on the tail of the other monomer, recognized by specific relay proteins inside the cell. Each such protein binds to a specific phosphorylated tyrosine, undergoing a structural change that activates the protein. Each activated protein triggers a transduction pathway leading to a cellular response. In this case, one of the relay proteins will bind to phospholipase C. The result is the same cleavage of PIP2 and IP3 activating a calcium ion channel on the ER membrane. Calcium ions are then released into the cytosol, the same response as in the G protein pathway.
The signal transduction pathway for glucose regulation begins with an insulin signal. The insulin signal binds to an insulin receptor which has two subunits. The top subunit is a receptor and the bottom subunit has tyrosine kinase enzymes. The insulin signal triggers tyrosine kinase activity in the bottom subunit. This kinase phosphorylates two enzymes, mitogen-activated protein kinase and PI3 kinase. The activation of the MAP kinase leads to completion of functions like cell growth and gene expression. However, the PI3 kinase pathway is most important for glucose regulation because it is responsible for the distribution of glucose for important cell activities. Thus, excess glucose from eating food is used up within the cell. Type 2 diabetes involves a defected insulin signal transduction pathway. As the insulin signal binds to the insulin receptor, the receptor does not recognize the signal and there is no transduction pathway or response. Thus, glucose is not used in the cell and blood sugar levels become very high within the body. Symptoms of type 2 diabetes are very mild, thus they're not very noticeable. Symptoms include things such as being very thirsty, peeing a lot, blurry vision, being irritable, tingling or numbness in your hands or feet, feeling worn out, and wounds that don't heal. There is no known treatment of diabetes, however, there are steps you take to manage type 2 diabetes. These steps include healthy eating, regular exercise, diabetes medication or insulin therapy, and blood sugar monitoring. These steps help keep your blood sugar level closer to normal.